Thank you, Anne-Sophie. Good morning to everyone. And thank you for the PhD committee for organizing this very, very exciting meeting. So let's go back to biology, and you want to build a house. So as you are a biologist, you need to hire professionals with different and complementary competencies. So you need a management team with engineers, foremen, but you need also workers with different skills and functions that will do the work, that will use the material to build the house. So you don't know how to do that. That's the first time, so you ask for CVs, and you receive a lot of CVs. But it's very difficult to judge a person upon a CV, because it represents theoretical competencies, theoretical skills. The problem is that you want to know more than that, because that doesn't tell you about the people's ability to work together, practical skills, ability to fulfill the expected task. So you need to find a way to gain more information about these people you want to hire. You want to know how they work when they are all together on site, when they are gathered together. What are you expecting from these people? You are expecting some coordination of the different steps required to build the house. You are expecting that they do interact when necessary, they do crosstalk, and they really are together with the same aim. Sometimes, when there are difficult tasks to accomplish, you expect from them some kind of cooperation. And also, when someone for any reason is missing, you want to know if someone else will be able to replace it and do the job replacing the people that is deficient. And eventually, you want to know that those people are really complementary to really achieve the task you want to have. So, okay, you decide to analyze some work they have accomplished together. So you really want to see what they have accomplished together. So you visit some buildings, you visit some house, and you can check if all the required rooms are here, if the job was done properly, or if there are some problems. So maybe here you can see that maybe the engineer was not good, maybe the guy in charge of the concrete preparation was not good, whereas you can see that the roof is okay, so some tasks were done properly, whereas others were not. So according to the building phenotype and characteristics, you can analyze if the management team and or the workers were deficient or proficient for the different tasks. Okay, and eventually your house is built, so now you can go back in your lab and you can do some biology. And back in the lab, you hear about very important cell mechanisms. So actually that was just a few days ago. And you learned that the Nobel Prize in chemistry was given to three researchers, Thomas Lindahl, Paul Mudrich, and Aziz Sankar, because of their outstanding work on DNA repair. And you don't know anything about DNA repair, but you think that must be a very important mechanism. Okay, so what is DNA repair? And you learn that it protects DNA against genotoxics, and these mechanisms are able to remove all sorts of DNA lesions to prevent mutation and to prevent cancer. And you learn also that there are some genetic diseases because of mutation in DNA repair genes, for example, xeroderma pigmentosum, that leads to skin cancer and neurodegenerative diseases. And you also learn that DNA repair is involved in resistance to anti-cancer therapies. So very important issues associated with the DNA repair defects. So you look to different publications, and you want to know a little more about DNA repair. And you discover that DNA repair is a very complex system. So cells, DNA, can be attacked by different genotoxics during life. For example, and these different genotoxics coming from the environment, but coming also from internal sources, like reactive oxygen species, due inflammation, for example. So those genotoxics can lead to the formation of very different lesions onto the DNA. For example, you can have double-strand breaks, typically formed by ionizing radiations. You can also have intra-crosslink adducts formed, for example, by chemotherapeutic drugs. Also bulky adducts, like those created by UV. And different kinds of small base modification, oxidation, alkylation, and also single-strand breaks. Nowadays, there are more than 100 lesions identified. And for each kind of lesion, there are some DNA repair pathways that have evolved to be able to remove all these lesions and replace the DNA, put normal DNA. And these different subpathways, they are called non-homologous enjoining and homologous recombination. These two repair double-strand break. Intra-crosslink repair repairs those crosslinks here, like that. Nucleotide excision repair repair the bulky adducts that induce double helix distortion. Mismatch repair mismatches. Base excision repair remove those small base modification. So very specialized pathways that are able to cope with any lesion formed. And those repair pathways, they involved hundreds of proteins and enzymes. And for a long time, those subpathways that have been represented as independent mechanisms. Now we know that it's not true, that the proteins of the different pathways, they do interact a lot with each other, and they crosstalk. So now you know that DNA repair is a very complex system that involves hundreds of proteins and enzymes, but that's also a pathways that are highly coordinated. They do crosstalk with each other. There are a lot of interactions, and there are also overlapping and complementary functions. Okay. DNA repair is not alone in the cell, and of course, it's under the regulation of signaling pathways, different kinases, different tyrosine kinase receptors also, and different soluble factors. Okay, so it's complicated, it's integrated into the cell regulation. But DNA repair involves enzymes. And that's a traditional way of presenting DNA repair and DNA. They are often considered as the workers of the cells because they do the job. They are active. So actually that inspires you. You see DNA repair mechanisms very differently. You see the workers here, and you see the management team here that decides who will do the job. So now you want to investigate DNA repair, and you have your experience with the house building. But what you want to do, you want to put the workers on site together with the management team. So what are you going to do to perform this? You want to model DNA repair. You want to give the workers and the management team the necessary material so they can perform DNA repair. So you develop functional assay with cell extracts. Why cell extracts? Because they contain the workers and the management team, all the required proteins and enzymes to do DNA repair. So this is called functional assays, and those functional assays or cell-free assays have long been developed to measure DNA repair enzyme activities. Actually, they have been existing for more than 30 years. The principle of the assay is that you take substrate. Substrate for a DNA repair enzyme, it's DNA with a lesion. You choose the lesion. So you prepare the substrate. You incubate the substrate with the cell-free extract that contain the repair proteins. So those proteins, enzymes, they do repair the lesion. And you can measure as endpoint DNA repair intermediate or the repaired DNA. So you can evaluate the DNA repair capacity of the enzymes for this specific substrate, for these specific lesions. And that's the phenotype of these enzymes. But DNA repair is complex and involves multiple enzymes and proteins. So you need to take this complexity into account if you want to develop systems to characterize DNA repair. There is a rich way to examine systems that was developed some years ago. That's multiplexed analysis. With multiple analytes quantification, you can get more information. It's also a comparative technology. That means that you have a relative comparison between the different analytes. You can use bioinformatics to analyze your results. You can do clustering. You can use clustering classification method to get the correlations between the different analytes, but also to have information on similarities between the different samples. So you can map the entire DNA repair network, and actually that's what we need. So now we have the format. We want a multiplexed analysis of DNA repair. Okay, so usually when you investigate DNA repair, you put one substrate in a tube, you put your cell-free extracts, so you gain information of the enzyme that will repair the substrate. But most of the time, there is a separation needed to separate the different species that are created. Now, if you put your different substrate on a solid support, on a microarray, you address your different substrates at different locations on the microarray. You can gain information on different enzymes at the same time, and in addition, there is no separation needed. So that means that you can enhance the throughput of your assay, and you can also save a lot of time. So what are the advantages of functional assays over just looking at DNA, RNA, or proteins? When you look at the DNA, that's like the theoretical skills of the DNA. That doesn't tell you how will the enzymes that are coded really work. In addition, there are a lot of regulatory mechanisms that can influence the function of the proteins at the different levels of the protein and enzyme synthesis. At the level of the DNA, you can have polymorphism, mutation. At the level of the RNA, you can have alternative splicing. At the level of the proteins, you can have post-translational modification, oxidation, etc. So for enzyme characterization, it's more interesting to look at the function than to look at the DNA. Functional assays integrate all possible regulations, defects in a single endpoint measurement. In addition, it also takes into account interactions between the different proteins. Okay. Now let's focus on what is excision synthesis mechanisms. These are the main repair mechanisms that take in charge base and modification. So when you have a lesion, it is recognized by some proteins, enzyme that will eliminate this lesion. And there are some polymerases that will copy the residual DNA. So we have two steps here. One that involves the cleavage of something, of DNA, and the other one that involves incorporation of the NTPs, and that means that you can take advantage of this step to label these reactions. Okay, so at Alex Repair, we developed the SPOT technology, and that's the functional analysis of DNA repair enzyme activities on biochip. The principle of that is that we put series of DNA lesions on biochip. So actually, we prepare them by synthesis. or we prepare them by irradiating plasmids, irradiating with UV, or doing some oxidation, etc. So we control the lesions. We prepare those series of DNA-containing lesions, and we put them on a biochip, and we incubate the biochips with cell extracts from samples we want to characterize. And actually, on the support, there are repair reactions taking place, and during this repair reaction, we incorporate or we eliminate fluorescent signals. So these assays, they are fluorescent, multiplexed, and quantitative, and at the end, we have what we call a DNA repair enzyme signature that characterizes your sample. Okay, and that's we work on glass slides with those little reaction chamber, and in each reaction chamber we have several substrates to be repaired by the enzymes. And this is Marie, and she's in the room somewhere here. So, what are the advantages of this system? The key features, of course, are miniaturization and parallelization. That's very difficult to say. So that means that you have many measurements at once. You have less consumption of reagent, of samples, and you can also use bioinformatics to analyze all that, so you have a more powerful and informative assay. Because of the biochip concept, because of the support, washing is made easier. On each spot, you have a local concentration of the substrate, so you have a more sensitive detection. And between the different pads, you have the same internal standard. So that means that you are more reproducible than reaction in solution. And that's amenable to automation. So actually, DNA repair becomes a system. And it's a whole new world to explore. Okay, so what can we do with that? In oncology, for example, you can stratify the patient's response to therapy, identify predictive biomarkers. That's very important to choose the best treatment. You can develop new drugs, for example, DNA repair inhibitors. You can check their specificity. You can control their mechanism of action. You can do aging study. DNA repair activities are really impacted by aging. You can maybe develop a protection solution. And you can also apply the assays to identify biomarker of exposure, for example, when you go into the sun. And you can also identify biomarker of risks. And indeed, that's very important in oncology to optimize the treatments, and in toxicology to develop protection strategies. Okay, so the assays were developed while I was in CEA. So this is a joint laboratory with the university. That's a laboratory of nucleic acid lesions. So Alex Repair was created to use these technologies mostly in oncology. We want to develop diagnostic tests. And our aim is really to bring DNA repair assays from research labs to the patient's beds, to hospital. I would like those assays to become routine analyses in the hospital. And I thank you for your attention.